Hi everyone, thank you for checking out our video. My name is Justin Clark and I'm going to talk to you about the bioinformatic analysis of therapeutic phages. So if you are on Twitter, I'm sure you know Sabrina Green, the mother of phages, our director of research and development. And if you've seen some of these other videos, you might have seen some of our other members, like Austin Terwilliger, the director of operations. But here at Taylor, we are a Bacteriophage initiative and service center at the Baylor College of Medicine in the Texas Medical Center in Houston. Now, we specialize in single-use patient cases, basically patients who have no other options but have a chronic creeping infection. So what generally happens is a doctor will get up with us because they think they have a patient who can benefit from what we do. So they send us a sample. We go through our library, which is always growing, and try to look for something that will infect and kill that bacterial strain. If we don't have it, we will search for things locally and try to find new phages or evolved phages or even engineer phages. Once we have a collection of phages that can attack that bacterial strain, we go through a whole list of preparation to try to make sure that that phage is suitable for use in a patient. We, of course, do the basics, the lytic parameters, sterility testing. We also do something called, we make something called synograms, which look for antibiotic synergy between phages and antibiotics. But we also sequence the phage, and sometimes when possible, the host, to make sure that the phages that we will be putting into a patient are suitable for human use. So that's really what I'm going to talk to you about today is what do we look for and what do we consider to be a red flag? So first of all, if you're here, you might already know about phages, but I always like to give a little bit of background on it first. Why do we like phages for treating antibiotic resistant infections? Well, they're easy to discover. Nature is almost an unlimited source of them. They're relatively cheap. They're evolvable, which is really important. With antibiotics, one of the main concerns is it takes all this time and money to create an antibiotic, and then maybe just one or two mutations later, a bacteria can become resistant. And then your antibiotic becomes much less effective. With phage, we have the advantage of being able to counter-evolve those. Bacteria will evolve resistance against phage, but the phage can then in turn evolve around that. And that's just a sampling of some of the reasons why we like therapeutic phage. What do we look for though? When possible, we look for high virulence of the phage, something that can infect rapidly, kill rapidly, make a lot of itself. Whenever possible, we'd love to have phages that can target virulence factors or things that make the bacteria able to survive within a host, like LPS or some kind of adhesion, maybe. The rationale there is that if a bacteria evolves around the phage, the easiest thing to do might be just to lose the virulence factor. If they lose the virulence factor, they can resist the phage, but then they can no longer infect as well. We also like non-transducing phage and lytic phage, and I'll talk about this in the rest of this presentation. And if possible, we would like to resistance to be rare and for there to be a high cost of resistance. We had a recent paper by Keiko Salazar I will bring up a couple of times because it's a really cool paper that just came out this past week. And it also kind of targets some of the things we're going to talk about. For example, she looked at the bacteria that evolved resistant to phage and then evolving phage against those bacteria that became resistant and found that a lot of the time when they evolved around a phage, the bacteria evolved around a phage, they lost something important that they needed for bacterial infections like LPS, and they were truncated, or attenuated rather, so they couldn't cause infection. But as bioinformaticians or data scientists, we're here, and this is where we're going to focus today. So you may be familiar with the lytic cycle and the lysogenic cycle, but I just want to briefly touch on this too. So the lytic cycle is really what we think of when we think about a viral, most people think of when they think about a viral infection or a phage infection. That is, the phage injects its DNA or its RNA in some cases into the bacterial cell. It replicates this DNA. It starts making a bunch of proteins. These proteins get put together into a new phage and makes a whole bunch of itself. Then it bursts out and the cycle starts over again. This is a virulent phage through the lysogenic cycle. The other possible life cycle is the lysogenic cycle. This is when the bacteriophage or the bacteriophage injects its DNA or RNA into a bacteria, and instead of immediately killing that bacteria and making more of itself, it kind of hides out and becomes part of the bacteria as like a mobile element. We generally think of this as being something that inserts itself into the genome or the chromosome. So it'll take its DNA, use an integrase to put itself actually physically into the bacterial genome, replicate with it through maintenance, is what that's called, until it gets a signal called induction, where it will then switch to the lytic cycle. Phages that can undergo the lytic and the lysogenic cycle are called temperate phages, and that's what we don't want for therapeutic use. We want phages that can only undergo the lytic cycle. And I just want to note that people generally think of the lysogenic cycle as being phages that put their DNA physically into the chromosome, but they can actually exist in other forms, such as a free-form plasmid or even linear DNA, which is rarer. In these cases, they act just like an extra chromosomal element where they replicate as the bacteria replicates until they get that signal, and then they go through the lytic cycle again. Now, the reason we want to avoid temperate phages is because they're known to carry antibiotic resistance genes. In fact, one method of moving mutations in one strain in the lab to another strain involves making a mutation in one strain that has an antibiotic resistance gene, and then using transduction to move that antibiotic resistance cassette to a new gene or to a new strain. So that's been done for, oh my God, decades. And another reason we don't like temperate phages is they can carry virulence factors and toxin genes. Some very famous toxins are actually carried on a phage, including Shigella toxin. If you've ever played Oregon Trail, you're probably familiar with dysentery. That is caused by a toxin found on a phage. It's also the same toxin that's found in enterohemorrhagic E. coli, EHEX. If you remember the famous outbreaks in Europe and the US at some fast food chains and other places. It can also carry things like the lamidhesin, which causes increased binding of bacterial cells to surfaces. Now, this is kind of like the surface rationale for not wanting temperate phages. These are reasons enough not to want a temperate phage. But there are some more abstract reasons why you wouldn't like it for therapeutic either. For example, a temperate phage can induce phage immunity, meaning that when a bacterial cell gets infected with a temperate phage, it can now be resistant to other phages. Obviously, something we don't want when we're treating the patient. And even kind of a more abstract idea is that once the phage integrates into the genome or the chromosome of the bacteria, it now has selective pressure to keep the bacteria alive. So instead of wanting to kill the bacteria and not caring about their health, it actually wants to kind of promote their health as long as it can because it's beneficial to them. Now, briefly touching on transduction, this is something we see with all phages called generalized transduction. Now, generalized transduction is really just when there's a mistake in the DNA packaging of the phage. What happens is the phage inserts its DNA, it goes into the lysogenic cycle, and it gets a trigger, some kind of stressor that causes it to be induced. And when it comes out and starts making more of itself, it makes a bunch of these. Actually, I should mention this is the lysogenic and the lytic cycle. So even the lytic cycle can, I jumped ahead to specialized transduction. In generalized transduction, lytic and lysogenic phages can undergo this. It's just when they make more of themselves, they make more of their phage capsule, all the proteins that actually make up the phage, and then package DNA in. Sometimes they can accidentally grab a piece of bacterial DNA. When that happens, this phage goes out and it can still infect because the DNA that's inside the phage doesn't actually cause it to bind or to inject the DNA. So all the proteins are there to cause it to put that DNA into a new cell. And then that DNA from a bacteria goes into another bacterial strain or another bacterial or bacterium. This is not great, but it's also pretty limited, and it's pretty limited with the damage it can do because it's possible to transfer an antibiotic resistance or a virulence gene, some kind of deleterious gene this way, but it's rare and it will only go between strains that are very closely related that the phage can both infect, and it doesn't propagate itself. It's like a one-off event. So once it injects that DNA, now it's bacterial DNA that's in that new cell, so it can't make more of itself. Of a little more concern is specialized transduction. This only happens with lysogenic phages or through the lysogenic cycle. This is when a phage integrates into the genome, and when it comes back out, instead of making itself only, sometimes it grabs a piece of the bacterial DNA as well. If this happens, it can package that with its own DNA into the phage. And now this is an infectious particle that can carry bacterial DNA and reproduce itself. This doesn't happen as often as you would think, and it's partially limited by the amount of space that's in a phage capsid. A phage capsid is evolved to only have a certain amount of space, and that space is basically evolved to carry the amount of DNA that it has. So when it picks up a piece of bacterial DNA, it generally has to lose some of its own DNA from some other part of its genome, probably the other end. If that happens and it loses something important, then it's no longer infectious, it can no longer replicate. If those genes aren't important, then it can continue the cycle and become something that can infect a lot of cells and make more of itself, which is much less than ideal. There is some debate about whether or not phages that undergo specialized transduction are really bad, because there's some concern that you can move antibiotic resistance genes or virulence genes to other species or to other strains, which is important, especially if you're treating something that's very virulent. You can move some of the things that make it virulent to new strains. But at the same time, we don't know how often this happens in the human, and it also may not be a huge deal if we're looking at like an isolated infection, say like a hip infection. So you have a joint infection, you add phage to it, even if specialized transduction is going on, the phage may not be able to get to enough other strains or other bacteria to actually do anything. But to be safe, since this is a newer field and we're still learning a lot, we're still avoiding transducing phages. And so that's recommended. Now, some of the questions we want to answer once we get our sequencing data back is, first and foremost, do we have a phage genome? Sometimes it will happen if there was a mistake in the lab or a mistake with the sequencing core or a mistake with the company you use. You can get mostly bacterial DNA. This has happened once before and with us, and we pulled out actually Staph epidermidis, which is a strain that's found in the skin. It was probably just an innocent mistake with someone putting a cap on or taking DNA out, and a piece of dead skin fell in or something like that. So it happens, but that's obviously your first step, right? Make sure you have a phage genome. Then we want to start asking questions that I went over, like, does the genome contain antibiotic resistance genes? Does it contain virulence genes? Does it contain markers for the lysogenic cycle? And also, and this kind of touches on the point I talked about before, do we have bacterial contamination? And the bacterial contamination one's kind of easy to answer. It's really how many of the reads you get back map to bacteria versus phage. So I'm not really going to talk about that much in this video. But there's a couple of things I want you to remember going forward with this presentation and with your work. One, the phage is on trial. We can't prove a negative, right? So we can't ever say for certain that this phage under the right conditions do not enter the lysogenic cycle. So in this way, the phage is more on trial, and we're trying to prove beyond a reasonable doubt or beyond all reasonable doubt that this phage cannot do these things that we don't want in a therapeutic phage. So in that way, we act as more of judges than scientists. The second thing I want you to remember is that redundancy is good. Whenever you do something one way, it's always best to do it a second way with a different method if possible. So the general next generation sequencing analysis is pretty straightforward. If you work with next gen sequencing, you have to follow basically this same pipeline to get an annotated phage or an annotated genome. You undergo quality control. You make sure your reads have good quality. You trim out bad reads. You trim out small reads. You trim out low quality reads, things like that. You assemble these reads together into a contig. You take that contig and you annotate it and look for genes. And of course, there are a lot of different things that go into each of these steps, depending on what you're doing, what technology you're using, what you need it for. So we're not going to go over the next gen sequencing analysis. This is something you probably can get a better overview of with other videos or somewhere else. And this is really just best practices, right? So we're mostly going to focus on what we do after we get an assembly. So you have an assembly. Awesome. Now what? What do you do with that DNA sequence? The first thing I always recommend is taxonomy. At the very least, I blast the sequence. There are other programs, other software that are better for this to get a more accurate picture of what you're actually looking at. But at the very least, some kind of database search to look and see what is closely related to your phage. This allows you to ask some really pertinent questions to what you're doing. For example, do these similar phages have similar sizes to your assembly? If you get a 50 kb phage contig and you compare it to its 10 closest relatives and they all are 45 to 55 kb, it's a pretty good indication that you probably have all the DNA of that phage. If instead the 10 most closely related sequences are all really closely related, but they're all 175 kb, then you need to go back and look at your assembly and your reads and see if you've missed anything, see if there's been any mistakes. The other thing you can do is once you have a list of closely related phages, you can start investigating those through literature search and ask questions like, are the most closely related phages lytic or temperate? Are there any red flags in those phages that would cause you to not want to use that phage in a patient? For example, if you had phage contig, you blasted it, you found that the 5 most closely related phages all contain a toxin that would make the bacteria more deadly to a human. That's kind of a red flag, and that means you can zero in on this toxin in particular and see if it's in your phage. It doesn't necessarily mean that you can't use your phage just because related species have something like that, but it does mean that you should look more closely and you should zero in on that and make absolutely sure that that's not also in your phage. And just to give you an example of this, one of our phages, just for a patient we pulled out and doing a blast search for relatives, we found the most closely related relative was about 70% coverage with 94% identity. And doing a literature search on that, we found that it was a Klebsiella pneumoniae phage and a Salmonella phage. And actually the researchers who isolated and sequenced that did kind of the analysis we're talking about today to look for integrases and toxin genes and things that we wouldn't want in a patient. And in this case, they found that their phage that's related to our phage didn't contain any of those things, which is good news for us. It obviously doesn't mean we can go take our phage and put it in a patient now, but it does mean that we're not seeing any red flags yet. The next thing I like to do is a gene family analysis. And again, this can at bare minimum be done with something like BLAST, some kind of alignment algorithm. But the question we're trying to answer here is, are there any antibiotic resistance or virulence factors in the sequence that we put together, our phage sequence? So you can, like I said, you can take an alignment algorithm of any kind, something like BLAST, and you can use databases such as the virulence factors database, Victor virulence factors, the Patrick virulence factors database. There are several publicly available databases like this that you can use to make sure that your sequence, nothing in your sequence matches anything in those databases. There are a couple of specialized algorithms you can use, like shortbread, which optimizes, which is the blast search is optimized for like shorter sequences. But in general, you'll be doing this in multiple ways and maybe even through your annotation. So I start with the gene family analysis with just the nucleotides, but I also do it with the annotation. Again, this idea of doing it more than one way. I also like to use BLAST, but also shortbread, because again, 2 ways are better than 1. Now, once you're done with that, you can move on to the assembly. There's some other things you can do with your nucleotide, your contig, like if you have enough reads, you can do a variant analysis, which I do like to do. But that's not something that's universally done and not something you really have to do. I like doing it because if I have the reads, I like to use them. And I also like to know just how many variants, how many mutations are found in the reads for my contig. In general, we do really deep sequencing. We send our phage off for standard bacterial sequencing. So when we get back, several million reads, which are enough to give 10,000x plus coverage on our phage genome. If we use that to map to our de novo contig, we can then do a variant analysis and look for really rare variants in that population. And I generally don't find, more than a handful of 1 or 2 SNPs, which is expected. But what I'm really looking for are insertions or rearrangements or something like that would kind of give me a pause. But at the very least, you want to do the things I've laid out here. So you don't actually have to do a variant analysis. It's just something I like to do, and I would recommend looking into if you have the time and the expertise to do it. After we're done with this, though, after we've got our assembly checked out, we want to do the annotation. Now, when people talk about annotation, they're really talking about structural and functional annotation generally. Structural annotation is just gene calling, basically looking for open reading frames on your assembly, on your contig. The functional annotation is the next step. Okay, we have these open reading frames. What do they do? And that's done with like a homology search. So looking at what is closely related to that and seeing if we can predict a function from closely related proteins. I'm not going to get really into the nuts and bolts of making sure you have good structural annotation. There's some videos through Texas A&M's Center for Phage Technology, a really great center with a lot of videos. I'm going to add them at the end, talk a little bit about their pipeline. But I would check out them and some other guides specifically on going through genomes and making sure that you have good, high quality structural annotations. All right, and again, not to harp on this, but at least two different methods are recommended. I don't like using just one set of annotation software or one annotation software. There are several free ones that are widely available and widely used: Glimmer3, GeneMark-S, Phenotate, which the Phage directory has recently done a video on with the maker, I believe. That's a phage-specific gene caller I would recommend. Now with functional annotation, I really, really like RAST. And, I guess I should put this at the beginning of the video, but none of these companies or none of these software makers are paying me. I'm just going through and talking about my experiences. So my opinions are my own. I probably should put that at the beginning of the video, but anyway, RAST-TK. I do like the RAST pipeline. It does have some additional add-ons that make it better for phage annotation. I believe it uses the Phantom database as well, which is a database that may have been, I'm not sure if it's still being kept up, but it does use that. And I also use Proca. Proca is a bacterial genome functional annotator, but you can also use it for phages. I will say one more thing about RAST. It does a gene family analysis. So that kind of takes care of looking at your open reading frames and your coding regions to make sure there's no antibiotic resistance genes or virulence factors there too. So that's very convenient. There's also Multifate 2, which recently came out. Multifate 1 came out a few years ago, and Multifate 2, I believe, was released just a few months ago, late 2020 or early 2021. It kind of does all of this for you. And those researchers state that they actually tried to make it for people who didn't have a data scientist on staff to do this themselves, to go through like every step and put all these pipelines together. What it does basically is uses all the software that I pointed out here, or a lot of it I pointed out here, and then compares them and runs it through and then gives you an output of gene comparisons. So I would recommend checking that out if you're just getting into this and looking for something that may make your life a little easier. Some other software you can use for functional annotation. This is more on the manual side of functional annotation. It really depends on what your phage is used for. If it's going to be a phage, it's going to be used a lot, something that may go into multiple patients or may go into a lot of research. It's recommended to go through and try to figure out what some of the hypothetical annotations are, because if you have a phage that isn't closely related to some of the more well-studied phages, it's not uncommon for 70% of the genes you annotate to be considered hypothetical genes or phage genes. If you want to resolve those, here's a list of software to kind of get an idea about what those things could do. It's not guaranteed that you're going to be able to pull out a function, but it is possible to maybe get a better idea. Maybe look for domains. For example, you may find some hypothetical proteins that have a DNA binding domain that may give you an idea about what it does. And I'm going to have all of these software up at the end when I talk about some questions I had from the last time I gave a seminar like this. Now, lastly, one thing you would want to look at is phage lifestyle. This is also done in the lab, doing like mitomycin C inductions to look for temperance or lysogenic phages. But there are a handful of software to try to predict this de novo. The ones that use nucleotide inputs are Phage AI, Phage Artificial Intelligence. This is a software that is web-based, and it supposedly uses, or it does use machine learning to look for association between temperate and lytic phages using available known lytic and temperate phages. So it predicts where your phage is and what it's most closely related to, and then it uses that to try to get an idea of what its lifestyle. Another one that recently came out was Backflip, and this is out of the University of Texas at Austin. I haven't tried this myself, but it is brand new, and I know that some people have already started using it with good success. So I'd recommend checking that out, especially once it's done with a peer review process. Now, the original phage lifestyle predictor was FACS. This one's been out for a while. People have really good luck with it. I believe the phage director also did a seminar on FACS with the people who designed that software. This is amino acid based. So you take your coding regions, you pull out your features, and then you load it through this. They also have a web server, but I believe they also have a local version you can run. And lastly, I do an annotation-based search for markers for lysogeny, such as integrase. If you have something that's annotated as an integrase in any of your annotations, that's a huge red flag. Probably means it's a temperate phage that inserts itself into the bacterial genome, and you're not going to be able to use it. You also want to look for mobile elements, because these can also tip you off for things like that. Or at the very least, it can reorganize in the bacterial genome. Excuse me. The other thing some people look for are recombinases, but you have to be careful with recombinases. Recombinases aren't necessarily integrases or something bad. In fact, an example of this is from our work too. We isolated a phage K-like phage that was very similar to the known phage K. And phage K is a lytic phage that's well characterized, and it infects Staph aureus. But our annotation of our phage K-like phage pulled out a recombinase in it, but it turns out this is actually probably a Rec A recombinase A, and others have annotated this as a DNA repair enzyme, and that's probably what it's doing. So if you get a recombinase, I always recommend looking into it more, but it doesn't necessarily mean that you have an integrase there. There are some other things you can look for too, like attachment sites. Faster software program that looks for prophages and bacterial genomes, but can also be used with phages, kind of like an off-script thing. You can put a phage in, and they do a blast annotation and look for attachment sites. They don't have all attachment sites, of course, but they do have some of the more well-known ones, so you can look for them that way. There are also things that are also proteins that are indicative of lysogeny, like resolvases, or I forgot what the other one is. It's a terminase, something like that. They can be indicative of the lysogenic cycle, but there's not enough known about them to try to predict them yet. But that is definitely something to keep your eye on going forward, to see if we can get a better idea and can better predict lysogeny. So now I'm just going to go over a couple of web-based software that I found useful. Again, I'm not affiliated with any of these labs or any of these websites, so I can't speak for them at all, but I can tell you that I've used them and I like them, and I can recommend you looking into them at least. One is Patrick. This is probably one of the easiest to use. It's geared more towards bacterial bioinformatics, but it does have a lot of software to analyze next-gen sequencing with pre-processing assemblies, several options for assemblies, and annotation. It does the RAST. It uses RAST to annotate, which means that it does the gene family analysis as part of that. It can also do a variant analysis, and it can classify your contigs. There's not a lot of options for customization. It's more, it holds your hand a lot, which is not necessarily a bad thing, especially if you're starting out or if what you're doing is pretty standard. But it's also the learning curve is not very steep on it. It's very easily used to get into. The next one I would recommend is Edge Bioinformatics. They're a lot like Patrick. They are a web-based bioinformatics suite. They have more options and more graphical outputs and more things you can do with it, including metagenomics. Although I believe Patrick may also do metagenomics, but don't quote me on that. They have a little bit higher learning curve because they have so many more options, but they do have a lot of really good software here. You can process most of your next-gen sequencing data through this. They have several assemblers For mapping, you also can annotate with PROCA or RAT. They do have a gene family analysis, and their gene family analysis uses the shortbread algorithm to search that I mentioned earlier. So I would recommend checking them out as well. I really, really like their taxonomic classification. They got a really in-depth classification setup that I recommend looking into. And the last one I'm going to talk to you about is the Galaxy server from the Center for Phage Technology. This is something they've been working on for years, and they just released a paper about this in PLOS Computational Biology I recommend checking out. This gives you a lot of options, and I really like the Galaxy software, or the Galaxy platform. It's web-based, so it's easy to get into. You don't need command line experience, but it lets you do a whole lot of things. And you can also add command line programs to it if you have enough experience. But you really can set up a lot of complex pipelines to annotate your phage. The learning curve is a little steeper than the others, but I think it's worth it because it kind of scales with your knowledge. You can start with it, you can learn it, and then as you get better and more confident with what you're doing, it kind of grows with you until you can even set up your own server locally and just run it on your own computer. Since they are the Center for Phage Technology, they're more geared towards phage annotation. So they have a lot more software geared specifically towards phage, like phage term, which looks at the read mapping to see if it can figure out what type of packaging the phage uses, like, well, you just look into it. It also has lifestyle prediction, facts, and backflip, as I mentioned earlier. And you can pretty much do everything I mentioned before, except gene family analysis. They don't have that integrated yet, but I believe they may be working on it, since you really only need the algorithm and the databases. But again, I'm not affiliated with them, so I can't really speak for what they're doing or where they're going. Just recommend checking them out. As far as tutorials goes, everything I just mentioned, the Center for Phage Technology, Edge, and Patrick, they all have guides. They'll have video guides that you can check out, which makes it pretty easy. There's also the phages bioinformatic guide, which is a really in-depth bioinformatic guide I would recommend. It's a little daunting at first, but it is definitely worth it. There's also a famous paper that came out a few years ago that I really like, called Categorizing Phage Genomes for Therapeutic Use. This is from the US Naval Research Institute, I believe. And they kind of laid out a lot of what I talked about today, and they used the CLC, which is, I believe, Thermo Scientific's commercial bioinformatics software, and the Edge platform, which is this is actually where I learned about the Edge platform, and I really, really like that. So of course, this is an academic video, so I got to give my acknowledgments. Thank everyone in the Morasso lab and the founders of TAILFR. But if you want to follow us, here's our information down at the bottom. And I definitely recommend also following Sabrina Green on Twitter, at Mother of Phages. And I'm going to leave this software list up as I go through some questions from the last time I gave a similar seminar. Just hopefully it'll help and maybe answer some of the questions you may have that I can't answer for you right now. But as far as the software list goes, most of these are free. I recommend checking them out, learning more about them if you're interested in using them. So feel free to look into that. All right, some of the questions I had from the last time I gave a similar seminar. My printer's a little screwed up right now, so it might take me a little bit to read some of these lines. All right, have you created a script to automate all of these steps, connecting all the various software tools, or do you do it manually? I would say I do it semi-manually, if that's not a cop-out. There are a lot of checks I do along the way, so I tend to bundle things together and then check them as I go. For example, the assembly, I have that automated, so I have it to where I can process all of that, and then I check my assembly before moving forward. Then I have the assembly automated, and then I have the finishing part automated. Right. Is sequencing required for therapeutic phage use? This is a tough one. I believe it depends on a lot of factors, not the least of which is how available sequencing is to you and your lab. In some places, sequencing is very available. Where we're at, we are very fortunate to have a very good sequencing core right down the hallway from us, and connections at some companies we can use. Other people are not so lucky. And phage have been used for decades, especially in the former Soviet Union, without doing sequencing. So I'm hesitant to say that you absolutely have to do sequencing for therapeutic phage use. I don't want to gatekeep here, especially if it's beneficial to people in areas that don't have easy access to sequencing. So it really depends, I think, mostly more than anything on your local regulatory agency and what the standard of care is there. Let's see. Do you select phages of different families of phages when you're making cocktails? No. We really only exclude phages based on how related they are if the sequence is almost identical or identical. The reason for this is that even a few SNPs can change how a phage bind. An example of this is from Keiko Salazar's paper that I had mentioned earlier. She looked at phage and bacteria and how they evolve and counter-evolve in a mouse and in the lab. And she found that bacteria will actually often truncate their LPS to get around phage, but this makes them avirulent. But then the phage can evolve to infect those bacteria. And what she did was sequence the phage, and I put this together, the phage that can infect the resistant bacteria. And what we found was that among other SNPs, the two that were probably the most important were SNPs and adjacent codons in the tip of the tail fiber. It just changed 2 amino acids right next to each other, and those appeared to be able to make it bind and kill that bacteria that was resistant to the original phage. So just 2 SNPs could be the difference between resistance and binding. So if we have 2 very closely related phages, but we have a few differences in them, depending on other factors, like how many other phages we have in the cocktail, how well each of those phages infect, things like that, it's kind of a judgment call. We don't exclude things from the same family, to answer the question. Let's see. Can we predict the receptor binding protein of the phage from genome annotation? And what is the best strategy to do that? As far as I know, not yet. Maybe if it's the the phage tail or the tail spike is completely identical to known phages that have been studied, it's probably going to be very unlikely. But if they are 100% identical on the amino acid level, it's possible to say they probably bind the same receptor. But again, as I mentioned earlier, a few SNPs in those regions can vastly change how the binding works. As far as predicting the phage binding the receptor without like additional information or homology information, I would say that's probably impossible right now. And it comes down a lot to our inability to predict the structure of these tail fibers. In fact, phage tail fibers are used in part to judge how well de novo structural predictions do, like different software do. There's, I can't remember what it's called right now, but various structural prediction software do this competition every year where they look to see which software can best predict the structure. And the hardest one is, I believe, always a phage tail fiber. And it's because a lot of these tail fibers are trimers, are homodimers, where they wrap in on themselves in a specialized environment, and that produces the 3D structure they need to bind a certain receptor. So it's very hard from this to predict what the receptor is just from the sequence. Excuse me. Now, let's see where we're at. If you were looking for a phage against the strain, if we just sent a bunch of sequences from all over the world, from just the sequence, how confident would you be that the phage would work against the strain? Not really confident at all. Again, a few changes can have a real huge impact on what a phage can infect. Like the related phage I talked to you about earlier, the Klebsiella pneumoniae and Salmonella phage. Does that infect E. coli? Does it infect Shigella too? I honestly can't say. And that may be even harder than predicting receptors, because just because a phage can bind doesn't necessarily mean it can infect. It's still got to be compatible with the internal machinery of the bacteria. So right now, I wouldn't be very confident. There may be some cases, again, with homology that we could use. For example, if some of those phages were known to infect like E. coli sequence types 131, and we were looking at a sequence type 131. Let's say, okay, if we have 20 of those, some of them may bind, some of them may work against this ST131. But even studies we've done in the lab and others have done show that a phage that can infect an ST131 can infect all ST131s. And we still don't know all the reasons why for this yet. So again, I wish I could give a better answer or a more optimistic answer with this, but as of now, no, not really. Let's see. Is it possible for a phage to appear lytic in the lab, but sequencing suggests that it is a temperate phage? Yes, actually, this is not super uncommon because it's both methods are not foolproof. To look for lysogeny in the lab, you infect a bacteria and then you stress it out. Mitomycin C treatment is a really popular one. There's also UV, there's antibiotics, there's I believe even hydrogen peroxide, but don't quote me on that one. Basically, you try to stress out the cell, and then you see if any of your phage come out after infection. This indicates that your initial infection, even if it lysed some of the cells, some went lysogenic, and now they're popping out when stressed. The problem is, obviously, the phage don't always respond to every stressor, and it's not really practical to test every phage against every possible stressor you can. What if some unknown stressor pulls it out? At the same time, bioinformatic predictions are based on what is already known. And so that's built on what we know in the lab. We have things like, for example, temperate phage and lytic phages can actually be really closely related, even more related than like lytic phages together, because they can be from different families or different genuses or different subfamilies, whereas lytic and temperate phages can be in the same family or in the same genus. So it's really hard to predict. And those are also all made based on associations. As far as I know, I have to double check on Backflip, but FACS and Phage AI are based on basically taking what is known, like lytic phages and temperate phages, and then seeing what your phage is more closely related to. Does it cluster with temperate phages? Does it cluster with lytic phages? That's the very basics of it. So that again is built on what we know in the lab. There's also the potential for those to be wrong, even if the data they're built on is correct. Say, for example, you have a phage that was temperate and all the ones that are similar to it are temperate. So if you put it in one of these programs, it's going to cluster with temperate phages. But then what if it's lost something it needs to go into the lysogenic cycle, such as an integrase? If it's lost its integrase, it may not go into the lysogenic cycle. It may just perform the lytic cycle, because recall that lysogenic phages can undergo both the lytic and the lysogenic cycle. You cut off one, it can still do the other cycle, but it may look, because of what it's closely related to, like it can still do both cycles. So yeah, this goes back to the idea that the phage are on trial. We're saying beyond a reasonable doubt, this phage is not going to do this or doesn't contain this. Okay. Is it possible? Oh, that's the last question. There was a 2017 ISME paper which says that phages rarely encode antibiotic resistance gene. What's your comment? I know the paper you're talking about, and this is not quite my field because I believe it's more based on the metagenomics of phages. But my understanding of the debate right now is that initially when microbiome really took off 10, 12 years ago, they were looking at the phage they pulled out of guts, and they found a lot of antibiotic resistance genes in those. And it might have also been other sources like soil and water, but they were finding a lot of antibiotic resistance genes in that metagenomic data. Now, I believe we understand kind of better what we're doing, and there's some question about whether those techniques and the analyses that were used are best for trying to predict antibiotic resistance, and it may have actually been bacterial contamination that was giving us the antibiotic resistance genes. Again, this is just kind of my surface knowledge of this issue. So my understanding is that at first we thought they carried a lot of antibiotic resistance genes, and now we're saying, maybe not. I can say that the dozens of phages that we've put together in the last few years, we haven't seen antibiotic resistance genes in any of them, which obviously our screens may be biased. We may be biasing against that, so I can't say for sure. I can't say if it's universal, but I don't think, I think it's still up in the air. That's the answer to the question. I think we don't know to what extent bacteriophage phages carry antibiotic resistance genes. I will, if I'm going to speculate though, because this is my video and I can, I would say that if there's selective pressure for it, the phages will do it eventually. Somewhere they'll do it. The question is, how widespread is it? Bacteria gain an advantage from having antibiotic resistance, so it stands to reason that phages that infect them may also gain an advantage to carry antibiotic resistance genes if they're temperate phages. So I definitely think it's possible. I definitely think it's something we should always look for. But as to how common it is, I can't speak to that. Okay, have you gone back and done genome analysis of phages already used in human therapy? No, but that is a great idea. I would love to do that if I had time. It would be very interesting to see which phages that now may be lytic or maybe predicted to be temperate, or we now know are temperate, or carry something that we would normally say, oh, that's a red flag, we shouldn't give that to people, whether or not those actually worked and how well they worked. I will say there is some danger of kind of like the acetaminophen problem. You know, when that first went through the FDA, standards were lower, so it was safe. But I've heard from many people that that's a medication that, given its side effects, wouldn't make it through the pipeline to become a medication now. So even though they worked back then, it doesn't necessarily mean that we should ignore the red flags now just because they worked back then. So it would be an interesting study, though, just to see, especially I think with temperate phages, to see how well they worked. And next, is there any pipeline or prediction method to screen phages in silico for target of a particular bacteria strain? I think I've touched on this. Yeah, I think I've already kind of gone over this. The answer is no, not really, not yet. Soon, hopefully, particularly with machine learning. Maybe that can pick out patterns that we haven't picked out. And there's additional parts of this question. How do you see the future of phage engineering with bioinformatic methods in this context? Wide open, really. Like I said, I think artificial intelligence may pick out patterns that we don't see that are going to be very helpful. I also think bioinformatics are going to allow us to critique phages a little more going forward. For example, we may learn that certain proteins or certain genes are not great for therapeutics, may interfere with it. We may learn more about how they make decisions for lysogeny versus the lytic cycle. Yeah, I think the field's wide open. I think bioinformatics is going to play a huge part going forward, and it's going to allow us to get a much better grasp on using phage for therapeutic use. If you had a limited choice, would you use phages with a lysogenic pass, but behaving as lytic with deleted repressor or other elements? Yes, if I had no other choice, and again, this is something for your local regulatory committee, but me personally, if I had no other choice to treat myself, I would be comfortable with a lysogenic phage that had good lytic parameters that had things deleted in it, like an anti-repressor or an integrator, something like that. I would be okay with that. Whether or not regulatory agencies agree with me with that is that's a little different. I think there's also I think it's also about assessing risks. All the reasons I talked about that you want to avoid temperate phages are because of the transduction or because of the selective pressure. Those are relatively minor risks when you're looking at a life-threatening infection. So if you had to choose between the possibility of limited transduction or the possibility of the phage giving some kind of selective advantage to the bacteria versus someone for certain dying of an infection, to me that seems like an easy decision to make. But again, there's a lot of factors. For example, if you gave a temperate phage because that's all you had to someone who had no other choice, are we worried about antibiotic resistance or virulence moving from that bacteria to other bacteria in them or to other bacteria out in the wild? And then this is going to depend on where the infection is too. If we're talking about like a gut infection or like a skin infection, that phage may have access to a lot of other strains and the environment. If we're talking about something internal, like an organ infection or like a joint infection, then I think the danger is less, and this is just my opinion. if those phages, those lysogenic phages picked up something nasty and then you excreted them, which you will do eventually, there are not going to be many of them. And then what are the chances that they're going to get to something else and cause something that couldn't happen in nature? I think I think the chances of that happening are very low. So I would feel more comfortable in cases like that. But again, these are the conversations that we're going to be having for the next few years. It's all about risk assessment. What should be the study path for a master's students to carry out research in this area? What courses and skill sets should they look to acquire? This is a tough one because it's not really my field to build, obviously, these educational systems. In my personal opinion, I would focus more on the phage biology and good practices and where to find more information about the phage, simply because you can't learn everything, right? No matter if you're a master's student or a PhD student, so you've got to go out and learn yourself. And I have found that since I was a molecular biologist, I was a bench scientist in grad school before I moved over to analytics at the end. I have always found it easier to get information about the computational side of things, the programming, the coding, than the biological side of things. That makes sense, right? If you need to learn Python, there are videos. There are tons of videos to learn this. There's apps, there's games to learn this. Not so much for the phage biology. Those are much more specialized. So in my personal opinion, I believe that if your time is limited, I would focus more on the things that are going to be harder to learn on your own. There are some published lytic phages, but according to FASTER and FACS database, those are temperate. Why so? I think this goes back to what I was talking about before with just, we can't be certain with either the lab methods or the bioinformatic methods. Basically, losing one gene can change what you see in the lab. Having one SNP can change what you see in the lab, and that can get propagated into the training set when people are putting together these lifestyle prediction software. It's not necessarily because someone made a mistake or someone did something wrong or someone's trying to hide something. It's just because it's messy and there's a lot of uncertainty. I would say that probably what happened is that the lytic phages were mischaracterized, or, well, I guess this is just repeating what I said, because it's possible that they were mischaracterized in the lab, but it's also possible that they were mischaracterized in bioinformatic methods because they actually are lytic, because they lost something, but they're closely related to temperate phages. So the bioinformatics software thinks they're temperate. Lab methods say they're lytic. It could also be the other way around. They could be lysogenic, temperate, but the lab methods didn't catch the stressors really to cause them to be able to find the bacteria or the bacteriophage to cause it to come out. So it may be going lysogenic, but they don't see in the lab because it needs some weird exotic stressor to make it pop out. And you can only really see it, as far as I know, when it's induced and it comes out of the bacteria. So the people in the lab may say, okay, well, this is lytic. We've done these tests and it looks lytic. So they call it lytic, but then bioinformatically it's related to other phages that are temperate, and it turns out to be temperate. So again, Phage On Trial. I think we're down to our last two questions, and I cannot read the first one. Actually, no, the last one. If anti-repressor genes or resolvases are present, can we consider it a lytic phage? So they're talking about other genes that are indicators for lysogenic cycle. I didn't talk much about them because not a lot's known. I tend to not pay attention to those. If I get something that's annotated as an anti-repressor or a resolvase, I check it out, but they're much harder to predict because we don't know nearly as much about them as we do with the integrases. So I would say, again, we're not sure. If I saw a resolvase or an anti-repressor in my genome, it would be a red flag to me. That would require additional research to double check that phage. And this kind of goes back to why I said that my pipeline isn't automated or manual. It's kind of semi-automated. I get these things and I'm like, okay, before we move forward, we see something like this, we need to go back and figure out what it is and look more into the literature and closely related genes. So that is all of my questions from the last seminar. And if you have any questions, feel free to email me or Taylor Labs. And I hope you will follow us on Twitter or Facebook or, well, I guess I think those are the only two. I guess our YouTube videos aren't up, but you're here, so subscribe. Anyway, hope everyone has a good day.